Good morning. I'm a polysaccharide, and this is the day in my life, metabolism edition. To start off my day as a polysaccharide, I need to undergo digestion. Here's a tour of the intestine. I'm going to get ready by breaking down my outfits, and hopefully I become a monosaccharide. Now that I'm ready, I'm going to go to the cytosol, where I hope the breakdown begins. See you outside. And by breakdown, I mean dance breakdown. There are 10 steps of glycolysis, and for each step, I do one dance move. And in each step, there's an enzyme that catalyzes the process. For example, hexokinase catalyzes the phosphorylation of glucose to glucose 6-phosphate. This creates ADP. Another enzyme, phosphofructokinase, catalyzes the first unique step in glycolysis, converting fructose 6-phosphate to fructose 1,6-biphosphate. This again produces ADP. Some steps are reversible, like this one with glyceraldehyde 3-phosphate dehydrogenase, which catalyzes the conversion from glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. So, I was able to redo my dance for my enzyme friend and produce NADH. Phosphoglycerate kinase is involved in an ATP generating step of glycolysis. In making ATP, it converts 1,3-biphosphoglycerate to 3-phosphoglycerate. Pyruvate kinase is the enzyme involved in the last step of glycolysis. This enzyme catalyzes the conversion of phosphoenolpyruvate to pyruvate. From this step, I am turned into 2 pyruvates. So, the net products of glycolysis are 2 NADH and 2 ATP. After glycolysis, me and my pyruvate twin tried recording a video, but our friend pyruvate dehydrogenase complex punched both of us, turning us into 1-acetyl-CoA and bringing us into the citric acid cycle.
Welcome to the citric acid cycle.
Okay, I'm in the citric acid cycle. This is the part of the day I'm not excited about, but I'll bring you guys to it. I'll show you guys how I do it. In this first part of the citric acid cycle, I turn into alpha-ketoglutarate, forming NADH and CO2. To turn into alpha-ketoglutarate, my friend NAD+ takes hydrogen away from me, forming NADH and forming CO2, turning me into alpha-ketoglutarate. In this next step, I turn into succinyl-CoA, forming NADH and CO2 once again. To turn into succinyl-CoA, my enzyme friend donates H+, forming NADH and CO2, thus creating succinyl-CoA. In this next step, I turn into succinate, forming GTP. To turn into succinate, I phosphorylate my friend GDP, generating GTP. In this next step, I turn into fumarate, forming FADH2. Hydrogen atoms are stripped off of me and donated to FAD, my friend, forming FADH2. This process turns me into fumarate. This next step, I turn into malate. In this part of the citric acid cycle, I combine with H2O, or water, to form malate. This next step, I turn into oxaloacetate, forming NADH. To turn into oxaloacetate, I release hydrogen atoms, converting my friend NAD+ to NADH. Each turn of the cycle produces 3 NADH, 1 GTP, 1 FADH2, and 2 CO2. Oxphos is my favorite part of the day because this is where I make the most ATP. It's very rewarding in the end. An electrochemical proton gradient is generated in the ETC, which is used in forming ATP. The first complex involved is NADH dehydrogenase complex. Since it has a higher affinity for electrons than NADH, it strips away electrons and forms NAD+. The next complex is cytochrome BC1 complex. Now my friend ubiquinone transfers me to cytochrome BC1 complex, pumping protons. The third complex is cytochrome oxidase complex. My friend cytochrome C delivers me to cytochrome oxidase complex due to increasing affinity for electrons. This process repeats until oxidase complex accumulates 4 electrons. We are now at ATP synthase. The passage of protons through the carrier causes the carrier and its stalk to spin rapidly, like a tiny motor. The rotation causes friction between proteins in the enzyme's stationary lollipop head, altering conformation and producing ATP. You just saw the process of metabolism occur in a span of 4 minutes, through glycolysis, the citric acid cycle, and oxidative phosphorylation. It's through these processes that we humans produce energy. In glycolysis, glucose or sugar was partially broken down by cells in enzymatic reactions. The citric acid cycle was a process that released stored energy through the oxidation of acetyl-CoA. Oxidative phosphorylation completes the breakdown of food molecules and generates the bulk of the ATP made by the cell. So why a day in my life video? This project is a way to inform younger audiences of the metabolic processes that occur in the human body. Although very complex, this process can be understood in even the simplest terms and the most innovative mediums. Metabolism allows for our bodies to function normally through homeostasis. Learning about metabolism and how your body burns energy or makes energy can help guide healthier food decisions and daily activity.